Okay, so we've talked about the plasma membrane, that double phospholipid layer with embedded proteins, and we know how substances move across it and which substances can move. So now let's move into the cell and look at the cytoplasm. Cytoplasm is everything between the nucleus and the plasma membrane. If you look at the cytoplasm, it's made of a intracellular clear viscous gel-like fluid that houses all the organelles. It's about 75 to 90% water and proteins. So when you look at a cell, um, you know that you have the plasma membrane, the nucleus, and the cytoplasm. So what is cytoplasm? Cytoplasm is cytosol and the organelles and inclusion bodies. So there's a table on your notes that includes the most frequently seen organelles in a typical cell, and we're going to fill this table in listing a little bit of the characteristics of the organelle and the function. So we're going to start with this organelle. This organelle kind of looks like French bread or kind of like a hot dog with curves inside of it. This is a mitochondrion. A nickname for this mitochondrion is the powerhouse because it provides the power for the cell. We go through cellular respiration, aerobic cellular respiration inside the folds of the mitochondria. So mitochondria will be found in high numbers in cells like skeletal muscle where we need a lot of energy. So in the cell, besides having mitochondria, you also have a series of membranes. We call that the reticula of the cell. Let's see why the reticula is important.
A secretory protein is synthesized inside the rough endoplasmic reticulum, migrates through it, and exits inside a vesicle. The vesicle travels through the cytoplasm to the Golgi apparatus. The Golgi apparatus modifies the protein. The protein is dispatched from the Golgi in a transport vesicle that then fuses with the plasma membrane, releasing the protein to the outside of the cell.
So looking at that membrane system, you see that there is a roadway that's attached to the nucleus. The roadway can be contain little granular organelles called ribosomes, and so if the roadway contains ribosomes, we call that rough endoplasmic reticulum, and then some of the endoplasmic reticulum is lacking the ribosomes, that's smooth endoplasmic reticulum. Rough endoplasmic reticulum has ribosomes on it. Ribosomes are protein factories. They are the organelle that makes the different structural and regulatory proteins in the cell. Once it makes that protein, you saw it in the previous film how it goes through the ER and it buds off in a vesicle and will go to the Golgi apparatus to be reworked before it's either used in the cell or sent outside of the cell. So we have smooth ER and rough ER. Rough ER contains ribosomes, and so on your chart, ribosomes are protein factories, makes proteins found in the cytoplasm and on the endoplasmic reticulum. So since this endoplasmic reticulum has the rough ribosomes, it's called rough ER. It's a membrane system with ribosomes, processes and transports proteins. It also can make some phospholipids. Well, what about the smooth ER? The smooth ER is also a membrane system going from the nucleoside into the cytoplasm, and it is without the ribosomes, and so what it does, it processes lipids and some carbohydrates. So it's a membrane system without, I didn't put the word ribosomes in there, but without ribosomes, and its job is to process lipids and some carbs. So remember the little movie we saw? After it moves through the endoplasmic reticulum, the protein or the substance that's made will then go to the Golgi apparatus. It's going to go in this side and then work its way and come out the other side. And this vesicle will either be contain some kind of substance we need inside the cell, or it will go and fuse with the plasma membrane and be released outside the cell. So this is the Golgi complex, or the Golgi apparatus. It's a stack of flattened sacs, the convex receiving side and the concave shipping side. And what it does, it modifies and packages substances for either transport out of the cell or use in the cell. Now, let's just say that one of these vesicles that was containing some substance contained hydrolytic enzymes that are going to break down a substance. Then we would call this vesicle a lysosome. Let's say that this one down here contained chemicals that are going to process hydrogen peroxide. This would be called a peroxisome. Let's say that another one contains food, then it would be a food vesicle or vacuole. So we really have to know what's inside of a vesicle inside a cell to be able to name it. To actually name a lysosome, I would have to tell you it had hydrolytic enzymes inside of it. So peroxisome, if it contains oxidase to break down free radicals and hydrogen peroxide. A lysosome, if it contains hydrolytic enzymes to digest. So looking at this picture, we see the rough ER up here making some proteins, and the vesicle leaving the rough ER and fusing with the Golgi apparatus, making its way through the Golgi apparatus, being processed, and then put in another vesicle. Now this particular protein is going to be secreted by the cell. Maybe it's going to be a hormone or something that's used somewhere else in the body. Notice that other vesicles can contain hydrolytic enzymes and then fuse with a phagosome that contains maybe a microbe that we've ingested from the outside, and those hydrolytic enzymes will then break down that microbe. So there's several different kinds of vesicles that we can make: lysosomes, peroxisomes, or we can make a secretory vesicle. So inside of your cell you have a cytoskeleton, a skeleton of the cell, and this is made of microtubules, microfilaments, and intermediate filaments. So this is the cytoskeleton. Microtubules are the largest. They form like a monorail system for transport in the cell. When that vesicle is released by the Golgi apparatus, how is it going to travel to the other side of the cell? Well, it's probably going to travel on a monorail microtubule, kind of like a one-track railroad. We also have microtubules that support and provide movement for the entire cell. In a muscle cell, you have actin and myosin, which will slide across each other and cause cardiac and skeletal muscle cells to contract or get smaller. Some of the cytoskeleton are called intermediate filaments, and usually these are seen as guide wires for internal support. When we talked about junctions between cells, we said that proteins could act as an anchor for structure inside the cell, kind of like setting up lattice work and something for it to stand on. So intermediate filaments act as guide wires for internal support too, and they have the ability to disassemble if the cell needs to be more flexible, and then assemble to give more support. So microtubule is a monorail for transport. This second one should have said microfilament. I'm sorry, that's a mistake on the PowerPoint. The second one should have said microfilament, and that's for movement. That's like myosin and an actin. And then the intermediate filament, those are the ones for support. So change that on your chart. It should be microtubule, microfilament, intermediate filament. Another or two other structures that you see in the cell that are associated with the cytoskeleton are centrioles. And in cells that have centrioles, you find two of them at right angles to each other. Notice that they're actually made of several microtubules in triplet. You have 1, 2, 3, 4, 5, 6, 7, 8, 9 arranged around a circle. These centrioles are important in cell division. We're going to see when we go through mitosis that they actually make up the spindles and the asters that help us to divide our sister chromatids or our chromosomes. The area where they're located is called the centrosome, and it should be near the nucleus. So two centrioles at right angles near the nucleus in the centrosome area, and they're important in cell division. Now a cell like a nerve cell or a muscle cell is not going to have any centrioles, and that should tell you that that cell is not going to divide. So another structure that we see associated with some cells is an extension of the plasma membrane called cilia. Cilia have the ability to move. They're short hair-like extensions. There are cells that line your trachea that have cilia that move mucus by it. You have cilia in your fallopian tubes and your vas deferens to help move gametes along. So the cilia we see in the human body doesn't move the cell, but moves substances past the cell. Another organelle that provides for movement is a flagellum. The only cell in the human body that has a flagellum is a spermatozoan or a sperm. And in the midpiece there is energy that causes this whip-like tail to go back and forth and propel the sperm once it's ejaculated. Another extension of the plasma membrane we see that does not provide for movement but does increase the surface area we've mentioned before, those are microvilli, and they're just extensions on the apical end of epithelial cells where we need to either secrete or absorb a substance. So now we're ready to move to the last part of our cell, which is the nucleus. In this picture, you see a big nucleus in the center, a dark staining spot inside the nucleus called the nucleolus. Now you will hear some people say nucleolus, but I was taught nucleolus. And then all this granular material around the nucleolus is chromatin. It's very thin and sand-like. So we know that our DNA is all spread out thin so that we can use it. You notice that the nucleus has a double membrane around it and that there are pores that lead from the nucleus out into the endoplasmic reticulum or the cytoplasm. These pores are large enough for RNA to come through, but so small enough that DNA cannot come through. And that kind of gives you a hint that when you're trying to make a protein, you have the pattern in the nucleus, but it can't leave. So we have to make RNA from the DNA pattern, and then RNA is actually what brings the message to all of these little ribosomes to make proteins. So we have the nuclear envelope that's double. We have a nuclear pore, big enough that RNA can go out, small enough that DNA can't. We have the chromatin, where the DNA is found. We have the nucleolus. This is where RNA and ribosomes are made. So let's fill that in on our outline. The nucleus is the largest oval sphere inside of a cell. Some cells can have more than one nucleus, then we say that it's multinucleated or it has more than one nuclei. An example would be a skeletal muscle cell that has many peripheral nuclei. We have masses of cells that go together and merge and in the embryonic stage, and so you end up with a multinucleated gigantic cell once they all go together. So there's a picture of skeletal muscle, and so this long fiber is considered one muscle cell, and you see those dark purple dots, those are all nuclei of that one cell. There is an couple examples of cells that have no nucleus at all in their mature state. One good example is a red blood cell. A red blood cell actually loses its nucleus before it comes out of the bone marrow and gets into the bloodstream. And this helps the red blood cell do its job more efficiently because now we have more surface area to carry oxygen. We're also not going to use our oxygen to repair or make energy because we've lost all our other organelles as well as our nucleus. The nucleus is important to a cell because that's where our genetic or our hereditary units are that control and regulate many of the cellular activities. So our DNA is found in the nucleus. Now we do have some mitochondrial DNA, but that's another lesson for another day. So most of the DNA we talk about in A&P is located in the nucleus and will contain the pattern for making proteins. Now your DNA is arranged in a single file in genes. You have 46 of these single file DNA pieces called chromosomes. When a cell is getting ready to divide, the DNA is going to be concentrated so that we can divide it properly. But when a cell is doing what a cell does, we need the DNA all spread out thin so we can get to the pattern. So you can see in this picture, this is DNA, a double helix, and then we wrap that double helix or that DNA around histone proteins to kind of organize it, and then we take it again and we wrap it on itself, and we're making it denser and denser and denser, and you can actually see the chromosomes under a light microscope, whereas you couldn't see chromatin, it would be too thin. Chromatin is when you're using it and it's all thin and spread out. Chromosomes are when we're going to divide it. We need it all concentrated in one place. So you have 46 chromosomes, and we call that DNA chromosomes when a cell is dividing, or we call it chromatin, and it has a very fuzzy appearance of tangled massive beads on a string, and the string actually would be the DNA. So thin chromatin is when the cell is using the DNA. Thick chromosomes is when the cell is dividing. So DNA goes from thin chromatin arranged around histone molecules, histone proteins, and when we want to divide it, we're going to wrap it on itself over and over. We're going to coil it and condense it so we can definitely divide it equally. So when you look at DNA, only 60% of the DNA 60% of the DNA are those organizing histone proteins. Only 30% is actually DNA, and then 10% is RNA chains that are being formed. We're using the DNA to make RNA. We actually have extended chromatin where we spread it out thin so we can use it in active segments, and then we condense it in non-active segments. So we're going to pull it out thin when we use it, and we're going to condense it back up when we're not using it. The DNA is found inside of the double nuclear envelope, and the pores can let large particles like RNA out, but DNA is just too large to get out. So we use DNA in the nucleus to make RNA, and RNA leaves. The nucleolus is a dark staining area in the nucleus where we make ribosomal RNA and ribosomes. So let's talk about DNA and what it looks like. DNA, if you remember from chapter 2, is a nucleic acid, and all nucleic acids are made of nucleotide units. And if you look down at the bottom of the screen, this is an example of a nucleotide unit. It's a phosphate, a sugar, and a nitrogen base. So a nucleotide unit is a ribose sugar, a phosphate, and a nitrogen base, and we arrange these so that they form a double helix or a twisted ladder. Now, inside of the ladder, the rungs of the ladder are where our nitrogen bases will be located. We have two types. We have purines, which are adenine and guanine, and we have pyrimidines, which are thymine and cytosine. Guanine, which is a double-ringed purine, has to link up or bond to a pyrimidine cytosine, which is a single ring. And so that leaves our double ring adenine purine to hook with our single ring thymine pyrimidine. So adenine always goes with thymine, A to T, and guanine always goes with cytosine, G to C. So let's look at this molecule of DNA. So this would be a long strand of DNA. And so we have phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base, phosphate, sugar, nitrogen base. This is one side of our DNA. DNA is doubled, so we have another chain of nucleotides coming up the other side, but notice they're going in opposite directions. This 5' tells us which carbon we're talking about. So this phosphate is hooked to the 5 carbon. This one is hooked to the 3 carbon. So notice how the 3 carbon is up here. So this one's going in the opposite direction. So if I hooked a pyrimidine guanine right here on this particular nitrogen base, if this is guanine, what is this going to be over here? Cytosine. And if this is cytosine, what's going to be over here? Guanine. And if this is thymine, what's going to be over here? Adenine. And if this is adenine, what's going to be over here? Thymine. And then if this is cytosine, what's going to be over here? Guanine. So A goes with T and G goes with C. So what we do when we read a DNA code is we actually read down one side of the rung of the ladder. So when I'm reading this one, it would be TGA, GAC, and that is going to be our code of information. So DNA is double-stranded. It has the nitrogen bases adenine, thymine, guanine, and cytosine. RNA is single-stranded, and instead of having thymine, it's going to have uracil. Let's listen to this little movie to get a good idea of the differences.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
So, how are DNA and RNA different? DNA is double. DNA has deoxyribose, where RNA just has ribose. DNA has thymine, where RNA has uracil. And DNA has to stay inside of the nucleus as long as the nuclear envelope is intact. RNA can leave the nucleus. So, now that we've talked about the three parts of a cell, the plasma membrane, the organelles that are in the cytoplasm, and the nucleus, now let's talk about some cellular processes. Every cell goes through a life cycle. And so scientists call the cycle of a cell, or the life of a cell cycle, from when it began, when it was created in mitosis, until it itself divides in mitosis. So once a cell is created, it's going to have a growth period, or a G1 period. There is going to be a checkpoint in there to make sure that everything's going on all right in the cell, making sure that anything that we need in order for that cell to duplicate is present. Then we go from the G1 growth cell, when the cell is doing what that cell does best, whether it's contracting or secreting or protecting. Then if it is able to divide, it's going to go into the synthesis, or the S phase. During this phase, you're going to have growth and DNA synthesis. This is where you're going to double your DNA and start making some of your organelles double, so when you divide, there's enough to go to each one of the new daughter cells. Then you're going to go into another growth phase, a G2. The final preparations for division are occurring here. You're going to have a second checkpoint to make sure everything's in line to divide properly. All of this that's in green, growth 1, synthesis, and growth 2, this is called interphase. They used to say interphase was a resting phase, but that's actually not true. That's when the cell is doing what the cell does best. What it's arresting from is it's resting from dividing. If I'm in a movie and we have an intermission, I'm taking a break from the movie. So interphase is taking a break from dividing. So after I have gone past my G2 checkup, then I'm going to go into the mitotic phase of the cell cycle. Mitosis is divided into 4 phases: prophase, metaphase, anaphase, and telophase. In prophase, you'll see certain things happening to the nucleus and certain things being created, and we're going to go through that in detail in just a moment. So this part of the mitotic phase, prophase, metaphase, anaphase, telophase, is when the nucleus is dividing. Well, we want 2 cells, so we also have the cytoplasm divide in cytokinesis. And notice that that starts in late anaphase. We'll talk more about this later. So a cell goes through G1, then it starts to prepare to divide by doubling its DNA, doubling all of its organelles, doubling its centrioles. All of that happens in interphase, and then finally you go into mitosis, which is cell division. So a cycle of a cell events that ends with cell division is a good definition of a cell cycle. There are 2 types of cell division. The one that we're talking about here with this cell cycle is mitosis. Mitosis is the production of normal somatic body cells. They produce somatic cells that have 46 chromosomes. But we also make another type of cell called a gamete, which only has 23 chromosomes, which has half of the homologous pair. That process to make gametes or sex cells is called meiosis. We're not going to mention meiosis or go through the steps. That's really something you do in a general biology class. I just want you to know that meiosis is the production of gametes or sex cells. And we start with a somatic cell, but we divide twice and end up with a gamete or a sex cell with a haploid number of chromosomes. So here's the cell cycle again. You can see all the phases of the cell cycle. So the steps in the cell cycle ending with mitosis. First you have interphase. Interphase is made up of G1 growth. This is when some of the centrioles start to replicate. Then we end up with the S phase. This is when the DNA replicates. And then finally, we're going to go into the G2 where you have your final preparations. So let's talk about DNA replicating first.
In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, A with T and C with G, each strand can be used as a template to build a new complementary strand, producing two DNA molecules. In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Eventually, all the replication bubbles merge, yielding two identical DNA molecules.
Okay, so how does DNA replicate?
In principle, copying DNA, a process called
So DNA replicates by unzipping, and we can see the nitrogen base pairs there. And so if what's being exposed is an A, we would build a T onto it. And if it was a G, a C, so that we end up with two strands of DNA. Each half of the DNA came from the original. So it's like we have two new ladders, and on each ladder we have one of the old sides and a new side. And then the rungs of the ladder, of course, are your nitrogen bases. So we went through the G1 phase, we went through the synthesis phase where we doubled our DNA. Then we'd go through some more division and preparation in G2, and finally we go through the M phase, which is mitosis. Not only are we going to divide the nucleus, which is actually mitosis, but we're going to also divide the cytoplasm. So we're going to divide.
Cell division consists of two processes, mitosis and cytokinesis. Mitosis, division of the nucleus and its chromosomes, is divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis is followed by cytokinesis, when the cytoplasm splits to form two separate daughter cells. The cytoplasm of a cell in late interphase contains two centrosomes, each of which may contain a pair of centrioles. In the nucleus, the chromosomes have been replicated during S phase, but are still dispersed in the form of chromatin. One or more nucleoli are present. During prophase, the nucleoli disappear and chromatin fibers coil up to become discrete chromosomes. Each chromosome consists of two identical sister chromatids, joined at the centromere. Microtubules grow out from the centrosomes, initiating formation of the mitotic spindle. During prometaphase, the nuclear envelope breaks into fragments. Some of the spindle fibers reach the chromosomes and attach to kinetochores, structures made of proteins and specific sections of DNA at the centromeres. Non-kinetochore microtubules overlap with those coming from the opposite pole. At metaphase, the mitotic spindle is fully formed, and the microtubules attached to kinetochores move the chromosomes to the metaphase plate, an imaginary plane equidistant from the poles. Anaphase begins when the two centromeres of each chromosome come apart, separating the sister chromatids. Once separate, each sister chromatid is considered a full-fledged daughter chromosome. Motor proteins of the kinetochores walk the daughter chromosomes along the spindle microtubules toward opposite poles, and the microtubules shorten. At the same time, the spindle microtubules not attached to chromosomes lengthen, pushing the two poles farther apart and elongating the cell. During telophase, nuclear envelopes form around the identical sets of chromosomes at the two poles of the cell. The chromosomes uncoil and nucleoli appear in the two new nuclei. Meanwhile, cytokinesis begins, splitting the cytoplasm and separating the two daughter cells. In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
So, you saw the phases of mitosis or cell division. So let's look at cell division or the phases of mitosis. So we're going to go through interphase where our DNA doubles and our centrioles replicate. And then when we enter mitosis proper, we were in prophase. So there are several things that happen. There are spindles that form between the two centrioles. There's asters that form around the centrioles. The centrioles move to opposite poles. The nuclear envelope starts to degrade. The chromatin starts thickening up to chromosomes. So by the time you get to late prophase, you have no nucleus, no nucleolus. You have visible DNA as chromosomes. The centrioles are at opposite poles with spindles in between them that are connected to the kinetochore of the homologous chromosome pair, and you have asters around the centrioles. So a lot goes on in this early prophase. This is probably the hardest phase of all the phases. So this would be early prophase and late prophase. Metaphase is the next phase, and it's pretty easy because the sister chromatids, which they're officially called now, line up on the equator or in the middle. So we line up in the middle in metaphase. Anaphase is pretty easy too, because in anaphase the sister chromatids are pulled away in anaphase, and when they're pulled, it's kind of like a worm being pulled on the end of a fishing line. They kind of go out into a V or a horseshoe shape. What you can't see on this particular picture is also cytokinesis starts here, so you should have a little bit of cleavage furrow on each side of the cell, like a little indentation on each side of the cell right in here, if this were a true representation of late anaphase. And then the last phase is telophase, and it's pretty easy too. You get two cells in telophase, but really it's really, how many times am I going to say really? It's really the opposite of prophase. Remember, in prophase, your nucleus, your nuclear envelope and your nucleolus disappeared. Well, in telophase they reappear. Remember that spindles and asters appeared in prophase. They're going to disappear in telophase. Remember that your chromatin thinned out into chromosomes in prophase. Well, the chromosomes are going to uncoil into chromatin, and then your cytokinesis is going to be complete, and you're going to have two separate daughter cells that are identical to the mother cell. Let's listen to it one more time.
Cell division consists of two processes, mitosis and cytokinesis. Mitosis, division of the nucleus and its chromosomes, is divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis is followed by cytokinesis, when the cytoplasm splits to form two separate daughter cells. The cytoplasm of a cell in late interphase contains two centrosomes, each of which may contain a pair of centrioles. In the nucleus, The chromosomes have been replicated during S phase, but are still dispersed in the form of chromatin. One or more nucleoli are present. During prophase, the nucleoli disappear and chromatin fibers coil up to become discrete chromosomes. Each chromosome consists of two identical sister chromatids, joined at the centromere. Microtubules grow out from the centrosomes, initiating formation of the mitotic spindle. During prometaphase, the nuclear envelope breaks into fragments. Some of the spindle fibers reach the chromosomes and attach to kinetochores, structures made of proteins and specific sections of DNA at the centromeres.
They're calling this
Non-kinetochore
prometaphase. We
microtubules
just
overlap
call it
with
late
those coming from the
prophase.
opposite pole. At metaphase, the mitotic spindle is fully formed, and the microtubules attached to kinetochores move the chromosomes to the metaphase plate, an imaginary plane equidistant from the poles. Anaphase begins when the two centromeres of each chromosome come apart, separating the sister chromatids. Once separate, each sister chromatid is considered a full-fledged daughter chromosome. Motor proteins of the kinetochores walk the daughter chromosomes along the spindle microtubules toward opposite poles, and the microtubules shorten. At the same time, the spindle microtubules not attached to chromosomes lengthen, pushing the two poles farther apart and elongating the cell. During telophase, nuclear envelopes form around the identical sets of chromosomes at the two poles of the cell. The chromosomes uncoil, and nucleoli appear in the two new nuclei. Meanwhile, cytokinesis begins, splitting the cytoplasm and separating the two daughter cells. In animal cells,
now, there are some exceptions to the steps that we just looked at in cell division. There are some cells that stay at G1. They never get to go past that G1 checkpoint, so we say that they're in G0 phase. Those are cells that don't divide, like skeletal muscle cells and like most nerve cells. And so they never go past that point, so they never divide. So once you've damaged a muscle cell, you've damaged it for life. There's no getting any more to replace it. Now, another exception to this normal growth cycle or the cell cycle is tumors. Tumors are considered uncontrolled cell division, and we have two kinds of tumors in the body. One that's malignant can metastasize and invade other parts of the body, and they interfere with normal functioning. So if you have a cancer or a tumor that's malignant, that's more severe than one that's benign, because benign ones are more isolated. They can be removed if they interfere with the functioning. They're not going to cause the systemic problems that a tumor that's malignant will. So let's go into now protein synthesis. Protein synthesis is a process in the cell where we make proteins. If you remember, ribosomes are protein factories. So this is going to take place in the ribosome eventually. This is where we're going to finally make the protein. But before we can make the protein, we have to have a blueprint or a template. DNA is the template for the making of proteins, but DNA has a problem. It can't get out of the nucleus. So what we do in the nucleus is that we use DNA as a pattern to make a messenger RNA. So this picture, you can see the DNA has unwinded, and we've exposed the nitrogen bases. We have a sense strand and an antisense strand. So the side that they're using is the sense strand, and we're getting that DNA code by matching up the complementary pair on the RNA. So for example, if I had AA, I would have to have UU on the RNA. Remember, RNA doesn't have a T. And then if I had a C, I'd have a G on the RNA. So we're going to match up the complementary pairs. RNA is similar to DNA but single-stranded and has uracil instead of thymine. Messenger RNA takes the genetic code, gets rid of some introns that do not code, splices the exons together, and leaves the nucleus. So the first thing we're going to do is called transcription. We're going to transcribe the message from DNA to RNA. So what we do is we make this long strand of messenger RNA. Well, some of the code is just nonsense. It's just a placeholder to help protect the code. So before messenger RNA leaves, we have to get rid of the nonsense. So the nonsense is introns, and what's going to exit the cell is exons. That's the real code. So before that messenger RNA leaves, we take out the introns and only the exons leave the nucleus. So transcription, very easy, is DNA to RNA. The second step of protein synthesis is translation. This is where we're going to bring the messenger RNA, we're going to hook on to ribosomal RNA found in the ribosomes. The ribosomal RNA is going to read every 3 nitrogen bases. That's called a triplet codon. And for each 3 nitrogen bases or triplet codon, transfer RNA will bring in a needed amino acid. Remember that proteins are chains of amino acids held together by peptide bonds. So ribosomal RNA reads messenger's pattern, and then transfer RNA comes and brings in the opposite to that messenger RNA pattern. So if the messenger RNA had UUA, then the transfer RNA that's going to bring in the amino acid would be AAU. So it's going to be complementary. So the transfer RNA brings in the needed amino acid. They're hooked together in the ribosome, and that's how we make a protein. We put together the amino acids like beads on a chain. Actually, the first code is going to be a starting code, and the last code is going to be a stopping code. So translation is RNA to protein. Now look at this picture. So here we have in the nucleus, we have DNA. We use DNA to make messenger RNA by transcription. But remember, messenger RNA has some of those introns in it. So we take out the introns and only exons are going to leave the nucleus. We go to the ribosome and see how it hooks onto the ribosome. So ribosomal RNA is in there, and transfer RNA is going to bring in the needed amino acids, and you're going to make a protein chain, a chain of amino acids. Transcribe DNA to RNA, translate RNA to a protein. So let's say that this is my DNA code. What RNA would I make to go with this DNA code? Well, what goes with T? A. What goes with A? Well, we're making RNA, so it has to be U. What goes with C? G. What goes with G? C. What goes with A? U. A, U. A, U. T, A, U. G, C, G, C, G, C. What goes with C? G. What goes with C? G. What goes with C? G. What goes with A? U. What goes with C? G. What goes with T? A. So we have our messenger RNA. The black ones, I'm going to tell you, are the exons. The red ones are introns. So before this RNA can leave the nucleus, we've got to take the red out. So the edited RNA looks like this. Every 3 of these nitrogen bases is a triplet codon. So we have 1, 2, 3, 4 codes here. So when we go to the ribosome and hook to the ribosomal RNA, transfer RNA is going to bring the needed amino acid and it knows which amino acid is needed because of this code that matches up. Remember what goes with A? U. What goes with U? A. What goes with G? C. So this code, UAC, means to start. And this code, GAA, means to bring in the amino acid leucine, and the next triplet codon means to bring in the amino acid tyrosine, and then the last one means to stop. So we have a protein that begins with leucine amino acid and ends in tyrosine. Scientists have studied these DNA triplet codon patterns, and they know each pattern what it codes for. Look at this. UUA is for leucine. Also, CUA is for leucine. There are like 23 of these possible amino acids. Proline, CCC. You see this one, UAA is to stop the protein, AUG is to start the protein. So they figured out what triplet codon codes for which amino acid. So let's look at it again. Here's the DNA molecule, right? Here we've unwound it and we've got it all strung out. So we have to transcribe or make messenger RNA. So what goes with T, A, what goes with A, U, what goes with C, G, and all the way down. Now this one we have already edited it. This one is all introns. So then the transfer RNA brings in the amino acid because it matches up the triplet codon. Brings in the next amino acid, the next amino acid, the next amino acid, the next amino acid, and so on, and then we stop. So this is our protein. It's made of these 1, 2, 3, 4, 5, 6, 7, 8 amino acids all strung together by peptide bonds. So first we transcribe and then we translate. So this is two movies I just want to play again. One's about membrane transport. The other is about the potential across the membrane, because these are two important concepts.
In order for us to use the molecules and energy we get from food, we must transport them across cell membranes. Transport of nutrients is essential for all cells, but we'll use cells in the small intestine to teach the principles of membrane transport. Let's zoom into the surface of an intestinal cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can move freely across the lipid bilayer. Notice that the lipid molecules move down their concentration gradient from where they are more concentrated to where they are less concentrated. This is an example of simple diffusion. Diffusion is a form of passive transport. It does not require energy from the cell, but it does depend on the concentration gradient, size and charge of the molecule or ion. However, most molecules cannot cross the lipid bilayer. Instead, molecules like the sugar fructose move into intestinal cells by facilitated diffusion, moving down their concentration gradient and through transport proteins. It also differs because it acts only on specific molecules, and the transport proteins can become saturated, reducing the amount of diffusion. However, facilitated diffusion doesn't require energy from the cell, so, like simple diffusion, it's a form of passive transport. The majority of water molecules cross the plasma membrane by facilitated diffusion, also called osmosis, through specific protein channels called aquaporins. Some ions, such as sodium and potassium, require transport against their concentration gradients. The sodium-potassium pump actively transports these ions from where they are less concentrated to where they are more concentrated. This process requires energy from the cell and is known as active transport. Energy from ATP is used to move 3 sodium ions, shown in red, out of the cell, and 2 potassium ions, shown in blue, into the cell. Because the pump uses ATP directly, This type of transport is called primary active transport. Another type of active transport, secondary active transport, also moves chemicals against gradients. It doesn't use ATP as a direct source of energy. Instead, it uses the downhill gradient from one ion or molecule to drive the uphill movement of another ion or molecule. For example, sodium and glucose move into the cell by secondary active transport in a process called cotransport or symport, because both substances are moving in the same direction. Sodium ions move down their concentration gradient created by the sodium-potassium pump, but because glucose concentrations inside the cell can quickly exceed those outside the cell, glucose moves against its concentration gradient. The cell moves larger materials, such as proteins, from the intracellular fluid to the extracellular fluid, using vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell.
See it moving
Cells
along the
use all these
monorail?
processes, passive and active transport, exocytosis and endocytosis, to move materials across their membranes. The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.